Today I'll be talking about chocolate making and the fermentation process that is associated with it. So quickly and briefly talking about history first, and then I will finally move towards my main topic, which is the fermentation. So if we talk about history, about 100 years ago, during the time of World War, these chocolate bars were not as popular as they are today. They were mainly used by soldiers back then, and it was mandatory for them to carry these bars. As you can see in the middle picture, that there are specific instruction for them that during the fight or battlefield they have to take small bites of this chocolate about every half an hour. So we can judge that back then it was more of an energizer for soldiers. And as you can see in this picture that it was named as the fighting food back then. And if you look at this box, this was the box that the soldiers usually carry. And it is the real picture, and it is about 103-year-old picture. And it's still kept in some army museum in the memory of World War. So this was all about the history. Now we are moving towards the modern day chocolate. And if we talk about today's chocolate, now we have many different kinds of chocolate, different types of chocolate and different varieties there. And here you can see the milk chocolate. And then we have this white variety, white chocolate. Then we have this dark one. And finally, we also have this fourth variety, which is unsweetened chocolate. So whether we talk about any kind of chocolate, they are all made up of mainly three components, which are cocoa solids, cocoa butter, and chocolate liquor. And if I specifically talk about the first one, the milk chocolate, it is mainly made up of cocoa solids and obviously milk as its name suggests. And then if we talk about the white variety, it is mainly made up of cocoa butter and it does not contain any cocoa solid. That's why its appearance is white. And then if we talk about the dark variety, it contains both things, the cocoa solid as well as cocoa butter. And if we talk about the last one, you may have noticed something here that this picture has something else. It's not cocoa, it's something else. I will be talking about their difference, the difference between cocoa and this word in the later part of my presentation. So moving toward the next thing, now I will try to explain chocolate making in simplest way possible so that everyone can understand. Now here we can see a cocoa pod. The farmers harvest this cocoa pod, and after harvesting this cocoa pod, they take it and they crack it open. And after cracking it open, you can see that here we have this white structure. They are resembling garlic pod, and these pods are taken out. These beans actually are taken out, and they undergo the process of fermentation. After the process of fermentation, you can see that their color has been changed. It's brown now. And then they are taken to the next level where they are crushed, where they are roasted, and their color changes to black, as you can see on the picture. Then by next step, they are finally converted into the final product, which is the chocolate. Now it's not a single step, the last one. If I talk about this last phase, it's not a single step, it's actually a series of steps. At first, we will look at those series of steps, and then we will finally talk about the main thing, which is the fermentation. So let me talk about the series of steps now. And in the series of steps, as you can see on this particular picture, at first we have to take fermented beans and then load them on this structure. And then the workers will open these bags and they will spread cocoa beans on this conveyor belt. And after doing so, you can see that these cocoa beans are now moving and they are reaching this chamber, and this chamber has coiled structure. And this chamber here is connected with this oven, and this oven is able to produce superheated steam. That superheated steam will reach this chamber via this connection, and they will be roasted. They will be instantly roasted because the temperature here is 120 degrees Celsius. So they will be instantly roasted as they are moving forward. Then comes the next phase. As you can see on the picture now, these roasted crunchy beans are moving downwards toward this container. This is not an ordinary container, it is actually a crusher or grinder. And this particular grinder will grind those beans, and by doing so it will separate the main thing, kernels, from the shells. Shells are useless. Now all the things will move forward, and you can see here we have this fan like structure. This fan will blow away the lighter weight shells to this container, and ultimately they will be wasted, and they will be separated as a waste, as you can see in this portion. Now comes the next phase. Now the kernels are moving towards the next phase. You can see these are the rollers. The solid kernels move between these rollers, and as they are moving, as the rollers are rolling, they will generate heat. The rollers will generate heat. Due to that particular heat, these kernels would eventually melt, and they will be converted into liquor or liquid form, which is and this step is known as liquor formation, as you can see on the screen. Now comes the next phase. Liquor will move towards this structure. This is actually a giant press. I think, yes, you can see now. This is a giant press. We know from our basic chemistry that it's like a piston. It can move up and down. And this, when applied pressure, will squeeze out cocoa butter from this liquor, and the remaining thing will move forward. We know that this structure here, this container here, is collecting the cocoa butter due to the pressure applied on the liquor. And this cocoa butter, if I talk about this cocoa butter, it's not useless. It's not a waste. It would be ultimately used to form different things like white chocolate. It can also be added to certain other varieties of chocolate. Now comes the next phase. If you have seen that we have removed something here, and now the next thing is that we have to add something back to the remaining liquor. Now we will add sugar and milk to this particular thing in a structure known as mixer. I hope you are getting my point. Then on the next phase, the whole mixer will be moved towards this evaporator. As the name suggests, it will evaporate any kind of moisture that is remaining or that is present in the mixture. Then comes the next phase. Again, we are facing rollers, but this time rollers are there to improve the consistency and texture of the mixture. And then you can see the material has been moved to this container, and we have these two chambers. These chambers are basically there to add certain flavors to the mixture, and after adding certain flavors to the mixture, the material would be moved to the next thing. This is important. This is an important one. This device is actually known as concher. That concher is there to generate heat. It uses heat, and it beats or rolls the material, as you can see in this section. And this concher is there to do an important thing. Due to all the 9 steps, the previous 9 steps, an unpleasant flavor develops in chocolate. To remove that unpleasant flavor, this concher is there to beat and use heat, and finally it will get rid of any kind of unpleasant flavor from the previous 9 steps. Then comes the next thing, tempering. Tempering is quite easy. Up until now, up until the 10th step, we just use heat. It was all about heat. Now comes the phase when we have to cool the material. So tempering is basically a cooling process, and due to this particular cooling process, chocolate gets its characteristic features like the shiny look and brittleness. And at last, the material will be put into these molds, and finally when the things are dried, they will be wrapped and the product is ready. I hope you got the series of steps. Now comes the main and important thing, which is fermentation process. If you look at this fermentation process, I will be only focusing on two main things, like how exactly this fermentation process is carried out, and when exactly this fermentation process takes place or starts. So to answer this question, I have to take help of this picture. As you can see, these 4 pots are closed. They are not open yet. But if we talk about the other 3, they are open. As long as these pots are closed, no microbe can start its activity, no microbe can even enter it. So the process of fermentation is not started. But as they are open, you can see here, as they are open now, they are exposed to air, and air, we know it has microbes, and due to those microbes, the process of fermentation will be started. And then comes the next thing. Another factor that can aid or trigger fermentation is the hands of the workers. If the hands of workers are unclean and they are using those hands to remove the beans, this factor will also aid the fermentation process. And then comes the next thing. If the workers are using unsterilized knife to open and remove these cocoa beans, they will also provide the microorganisms. And the last thing is this unwashed basket. If they are using any basket that was previously used for fermentation and they are using the same basket for the current batch, then it will also provide the microorganism and trigger fermentation. But there is another factor as well. These cocoa beans are spread on these banana leaves, and these banana leaves contribute many beneficial yeast and other microbial agent to all the beans. So you can see this could be the fifth factor. And what exactly are those agents that are responsible for carrying out fermentation? These are actually our favorite LAB, which is lactic acid bacteria. Then we have acetic acid, then yeast, then we have this filamentum, and then we have this aerobic endospore forming bacteria. So all these agents are triggering fermentation. Now this is the actual picture. If we open that white bean from the inner portion, it looks like purplish or brownish structure. Quickly, I will try to give you idea about fermentation by using this graph. So I'm taking this graphical approach. If you can see, we have all the five members or agents that are causing fermentation. We have yeast, we have LAB, we have acetic acid, the spore formers, and we also have fungi or molds. So this graph is quite easy. If you can look at the bottom, we have days. It indicates that normal fermentation in the case of cocoa takes place almost 8 to 9 days. And all these agents perform their duty at specific time. If we talk about yeast and lactic acid bacteria, they start their work immediately. And yeast, if we specifically talk about yeast, it usually dies or performs its duty within the first 2 days of fermentation, and after that it dies. And then comes the lactic acid bacteria. This lactic acid bacteria works for or takes part in fermentation for almost 3 days. Now this acetic acid bacteria does not start its work immediately, but it takes a day to start its role and then ends at almost 4.5 days. Then comes the spore formers. Now you can get the idea that it starts its work from 4 days and up until the completion of the fermentation. And same goes for the molds or fungi. They start their work at 6 days and they complete their task on the completion of fermentation. Now, what exactly are they doing? What kind of substances are they producing? The yeast and LAB, they are contributing or producing ethanol, and ethanol would reach its peak at the first day. And then comes the lactic acid. We know we have LAB or lactic acid bacteria. Both the first two agents are contributing or producing lactic acid, and lactic acid will hit its maximum amount at the second day. Then comes the acetic acid bacteria, which produces acetic acid, and it will be maximum at the third day. And while all these while all these agents are performing their duties, obviously they are raising temperature, and temperature will reach peak, or the maximum temperature would be attained at somewhere between the point where half of fermentation is completed. So 4.5 days you will get the maximum temperature. All about this graph. Quickly, now we can talk about the ABC of fermentation, the three steps A, B, and C. So at first you can see this is the same bean with white pulp, and this white pulp would be degraded first. How? Yeast will start an anaerobic step. It will convert sugar to ethanol, And by doing so, you can see that in the form of droplets, this pulp is degrading. This is actually called sweating, like the way we sweat in warm temperature. These things are called sweatings. The pulp will be degraded due to the yeast and the conversion of sugar to ethanol. Then comes the next phase, which is acidification. If you look at this picture, now most of the white pulp has been degraded, but the next phase is we have ethanol here, and the ethanol would be converted to acetic acid by some other agent, and this would be the aerobic step. And you can see now pulp has been degraded, but there is this drastic change. Now the pulp color is bit light brown, and the sweatings are also there. And in the final step, it will, when all the agents have performed their work, the final cover of or the layer of this particular bean would be brownish, as I have shown you in the start of my presentation. So that was the ABC behind the fermentation of cocoa beans that is involved in chocolate making. I hope you got this idea. Next comes the difference that I was talking about. You can see cocoa is quite different from this other word cacao. Now, if I first talk about cocoa powder, if the beans are both fermented and roasted, then that particular process would lead to cocoa powder formation. But if the beans are just fermented and not roasted, that particular powder would be cacao powder. So you can get the idea. In one process roasting is involved, in other roasting is not involved. And you can also get the idea, if I talk about cocoa powder, there would be high temperature, and to process the cacao there would be low temperature. So there comes the next slide. I'm almost moving to the conclusion of my presentation. Now comes the toxin. There is a toxin within chocolate. It is surprising to hear, but it's true. There is this compound called theobromine. Now, this theobromine is quite toxic for animals, organism, but for humans, the dosage or the toxic dosage is around 1000 milligram per kg of the body weight. And the chocolate does not contain that much amount of theobromine that could be harmful for us. So we can easily and safely consume this chocolate without any problem. But if we talk about this particular animal, the dogs, you must have heard that dogs can't really process, or dogs can't really eat chocolate. Why? Because their metabolic system is quite slow to process this theobromine. We as human can metabolize theobromine at a good pace, but these dogs can't. So as their process is slow, they start to build this toxic substance within their body, and they ultimately die. Comes my second last slide, which is chocolate, a mood food. This chocolate, earlier we talked about chocolate as fighting food. Now we know that it is also a mood food. How so? Because it gives, or it contains tryptophan, an amino acid that can increase or produce serotonin in our body. And this particular serotonin is a hormone or neurotransmitter that can calm us down, and it's a natural antidepressant. Instead of taking the synthetic antidepressant, we can go for this chocolate, which is a natural antidepressant. And then comes some other health benefits. If we talk about some other health benefits, we know that at the start I told you about different certain types of chocolate. Out of all those chocolate, dark one is better. Why? Because it contains flavonoids, and flavonoids can help us to reduce diseases like cardiovascular diseases, because it contains more quantity of flavonoids. But the problem with our generation is that we go for taste rather than the health benefits. That's why we tend to use the milk chocolate rather than the dark one. And then the chocolate also improves our blood pressure and also the functioning of platelets. That's my last slide now. I am finally moving towards my conclusion and the current research. Now there is this myth associated with chocolate, that chocolate is bad for teeth, particularly for children. It could result in tooth decay and so on. But you will be surprised to hear that this CBHE or this chocolate or the cocoa beans are actually providing something good that can remove plague. Now this CBHE is a cocoa bean husk extract that can be used to form a mouthwash, and that is quite good against removing the plaque that particularly builds up in case of children. Now there is this next technique which is DGGE. This is actually a gel electrophoresis technique. The full form is denaturing gradient gel electrophoresis. It is used to judge the microbial changes that are taking place while the process of fermentation is going on in cocoa bean. Now the next one is quite interesting. Researchers have found that even the waste of chocolate, even the chocolate waste produced by the chocolate factory is useful. It gives us something good, it benefits us. When we feed that particular chocolate waste to E. coli, the bacteria, in turn it gives us hydrogen, and that particular hydrogen can be used to power up this fuel cell, and the researcher was successful to power up this fuel cell, and they were able to power up a small fan by using that particular hydrogen. And finally, my last point is that if the fermentation is carried out using a hybrid yeast instead of the normal one, you can see the name of the hybrid yeast, it is Cloviromyces. This Cloviromyces is found to yield better quality chocolate. So with that, I'm finally concluding my presentation. And if you want to look into the detail of these researches that I just talked about, you can search these links or the research papers, and you can get the detail in your spare time. Thank you for your patience. Thank you for listening. I hope you have learned something new today. Thank you.